Hello, my friends, and welcome to our video lecture on topic D1.3, mutation and gene editing. Today's going to be a lot more about mutation. We're not really talking about gene editing until we get to our additional higher level content. Our guiding questions for today: how do gene mutations occur, and what are the consequences of gene mutation? Our objectives: we're going to define what even is a mutation. We're going to talk about the different types of mutation, describe the consequences of mutation, and then outline some of the causes of mutation. That first objective again was to define mutation. Mutations are structural changes to the genes. We're changing the DNA molecule. We're changing the order of the nitrogenous bases in those genes. Here we have a tiny little piece of the gene that codes for the protein hemoglobin. Hemoglobin carries oxygen around on red blood cells. One tiny little mutation, one little change to this gene flips that A and T to a T and an A. That tiny little mutation, that change in the DNA sequence, is going to cause that protein hemoglobin to have a different shape, and that different shape leads to sickle cell anemia. Mutations happen a lot all the time, but we have a lot of mechanisms in place to fix them before they cause too much trouble. One of those mechanisms actually happens during DNA replication. Remember, we talked about DNA polymerase III, and one of its jobs was to proofread the DNA right after it was copied in replication. Another mechanisms that we have to repair the DNA, we talked about very recently in our gene expression lecture, where we can phosphorylate our nucleosomes. Remember the nucleosomes, those histone protein octets, and the DNA that wraps around them. We can add some phosphate groups, phosphorylate those nucleosomes to repair DNA breakage so that we don't have those mutations lingering in the genome. Mutations happen a lot. They happen randomly. Many of them we fix. Some of them, though, do persist in the genome. And we're going to talk about those ones that persist. There are lots of types of mutations. We are going to talk about just a few today. The first one that we're going to look at is what we call an insertion mutation, because we are literally inserting a nucleotide into the DNA sequence. Here is our original DNA sequence. Here is our mutated sequence. I've gone ahead and chunked our DNA nucleotides into triplets because these pieces of DNA are going to code for triplets of messenger RNA, and those triplets of messenger RNA are codons. When we insert a nucleotide into the DNA sequence, what happens is we shift the reading frame of our DNA to messenger RNA to codon plan. And so we call these insertion mutations also frame shift mutations. These are my reading frames. That DNA is going to code for a messenger RNA triplet. Again, that's a codon. That codon codes for a specific amino acid. When I insert one nucleotide, what happens is my triplet is now here. The next triplet is here, the next triplet is here, the next triplet is here. And so I have shifted my entire reading frame. And so my triplets of DNA to messenger RNA to codon, they're different, hence frame shift mutation. These guys are usually pretty serious in terms of effect because we're going to have a great number of amino acid changes to the protein. Similar but opposite is the deletion mutation. So here we have deleted a nucleotide from the sequence, again, original and then mutated. This is also a frame shift mutation because my triplet is now 1, 2, 3, 1, 2, 3, 1, 2, whatever comes after that 3. And so again, we have a frame shift mutation. It's going to cause lots of amino acid changes, and that's usually going to be a very damaging kind of mutation. Here we are looking at what is called an inversion. So I've taken these 3 DNA nucleotides and we just flipped them around, literally inverted, reversed them. If we invert a triplet, then we're going to keep the same frame shift here. Not going to have too many big differences. We could have longer inversions that might have bigger effects, but not nearly as bad generally as those frame shift mutations where the entire reading frame is messed up. And the last type of mutation that we're going to look at is a super common one. It is the base substitution mutation. Base substitution, we're literally just substituting one nitrogenous base for another. I had an adenine, now we have a thymine, we just flipped out that one guy. This is not a frame shift mutation. Notice that the integrity of those triplets is maintained. So just that one tiny little base is changed, and maybe sometimes this doesn't have an effect at all on our proteins, and sometimes it has really big effects. And we're going to talk about those consequences of mutations right now. We have talked a lot about sickle cell anemia as an example of a mutation. And again, we have our normal DNA sequence gets changed, that base substitution mutation. That leads to a different messenger RNA transcript. That leads to a different amino acid in our protein. That causes the hemoglobin to clump up, which causes those red blood cells to sickle, and people have sickle cell anemia. So this is an example of a mutation that can have a pretty serious effect. Other mutations might have more serious effects, and some have far less serious effects. Depends on quite a few factors. One of the factors is if the mutation is in a coding region of DNA or a non-coding region of DNA. Coding region, pieces of the DNA that actually code for proteins, like this mutation that leads to sickle cell. Non-coding regions of DNA, those mutations might not have a super big effect. If we're talking about DNA of the telomere, a mutation there is not going to have a big effect. Remember that telomeres are the very tips of the chromosomes. But we could also have mutations in the non-coding regions that are promoter regions or enhancer regions, and that could have a big effect on gene expression. We could also have changes in the messenger RNA transcript that have different degrees of effect. We're going to talk about some of those different degrees of messenger RNA transcription effects next. And first up, we're going to look at some of the consequences of frame shift mutations in the coding regions of DNA. So we're talking about the DNA that is transcribed into some messenger RNA. That messenger RNA is then translated into an amino acid sequence. Frame shift mutations come in two versions. We have insertions and deletions. Both of these can shift the reading frame of our DNA and then messenger RNA transcript. Hopefully you're looking at this and going, aha, this one's an insertion because we inserted this guanine into the DNA sequence. So here's my mutated sequence. We still have TAC, we still have AUG, we still have methionine as our start codon because we start with AUG, reading our codon chart, A to U to G is methionine, that's our start codon. We should have ATA to UAU to tyrosine. Now I have ATG to UAC. UAC, interestingly, also codes for tyrosine, so we don't have an impact to our amino acid sequence yet. AAG is now AAA, interestingly still phenylalanine. But my serine now becomes glutamine, and my glutamic acid is now, whoa, it's an early stop codon. So this one has a huge impact on the protein because I should have this guy going on and on for hundreds, if not thousands of amino acids to build that functional protein. Here, I only get 4 amino acids and then it stops. Definitely going to have a big, serious, huge impact on the protein. Frameshift mutations tend to have pretty serious consequences when they happen in coding regions of the DNA. Substitution mutations don't always have as serious an effect on the resulting protein as do those frameshift mutations. So here we have a single change, one base substitution. We went from guanine to adenine, which changes our messenger RNA from UUC to UUU. But what's interesting is that my UUU codon codes for phenylalanine, as does that UUC. And so even though I have a different codon, I have the same amino acid. And that's because our codon chart is what we call degenerate, which I don't know, has a negative connotation, I think, but just means that there are some repeats. So my UUC and my UUU codons both code for phenylalanine. So even though I have a different codon, I have the same amino acid. My protein is going to be exactly the same, even though there was a mutation. We call this type of mutation a silent mutation or a same sense mutation because we have literally the same amino acid being coded for, even though our codon was different. No effect at all on the resulting protein. And here we have another example of a substitution mutation. We had ATA. We flipped out that A for a T, so we just changed that one guy. The rest of the DNA sequence is exactly the same. So I've got this messenger RNA should have coded for tyrosine. Now my new messenger RNA codon codes for what? UAA is a stop codon. We have an early stop codon. What does that mean? That means that my ribosome is going to detach from this messenger RNA. And even though I've got lots of messenger RNA that could get coded into a protein, it's not going to. Literally, stop means stop. Even though I've got messenger RNA, there will be no more amino acids added. And so we cut off our protein quite early. This is going to have a giant impact, probably a negative one on the protein. Clearly, because we didn't even make it. The previous slide we looked at a silent mutation had no effect. This one is called a nonsense mutation. Nonsense mutations are base substitutions that come with early stop codons. And sometimes the stop codon, no, most of the time, the stop codon is not the second place in the whole chain. It can happen maybe halfway down, but even half a protein is not going to be a functional protein. Nonsense mutation, early stop codon. The one that we looked at on the previous slide, silent or same sense, no effect because we had the same amino acid. Let's look at one more type of substitution. And this particular substitution mutation is known as a missense. So again, we're changing just one base, so a base substitution from T to A. That's going to make our codon go from GAG to GUG. And then we end up with, instead of glutamic acid, valine is in our polypeptide chain. This happens to be the particular mutation that leads to sickle cell anemia. So if this is the mutation in the hemoglobin gene, and we end up with valine in the hemoglobin protein instead of that glutamic acid, then that hemoglobin is going to be clumpy and we're going to have sickle cell anemia. Every once in a while, though, most mutations are bad. Most mutations are not good. Every once in a while, we can have some super lucky mutations that change the protein just a little bit and make it better. And then, assuming that that mutation even happens in an egg or a sperm cell and get passed on to an offspring, then we have offspring that are more fit, they're better able to survive, they're more likely to pass this mutation on to their offspring, and then we have some evolutionary benefits. It's super important to understand mutations happen all the time. Not because we living things need the mutations to happen, they just happen. And every once in a while, in a very, very infrequent way, every once in a while those mutations are lucky and they make us, if they're in our egg or sperm, make our offspring better able to survive and have babies, and then we've got that evolutionary benefit. We do not mutate because we need to. We mutate randomly, and every once in a while, it's kind of lucky. These base substitutions can also be referred to as SNPs, or single nucleotide polymorphisms. Single nucleotide, because we're talking about just one A or T or C or G in the DNA. Poly is many, morph is form or shape. And so we've got many different forms of the ATCG in this particular spot, in these different individuals. What's super cool, these SNPs in genes, these single nucleotide polymorphisms, these base substitutions, lead to, in the coding regions of DNA, the formation of new alleles. So when we talk about genes and we talk about those different alleles, and some are dominant and some are recessive, or maybe they're co-dominant, these different alleles are formed through mutation. Some of them are good and some of them are bad. All of them are going to be a source of genetic variation within a species. Some of them are good and some of them are bad. I know I just said that. We're going to talk about an example of one that's not fabulous. Mutations in the BRCA1 gene. So BRCA1 stands for breast cancer type 1. A mutation in this gene is going to make us more likely to get not have to get, but more likely to get certain forms of cancer. The function of this BRCA1 gene is to phosphorylate broken DNA. Remember in our gene expression lecture, we talked about how our nucleosomes, those histone proteins in the DNA that wraps around them, those nucleosomes can be methylated if I want to shut down transcription. We can add acetyl groups if I want to open up the DNA and get it transcribed more frequently. We can also phosphorylate those nucleosomes when the DNA is broken, and that's going to help us fix that broken DNA. If there's a mutation in the BRCA1 gene, then we're not phosphorylating the DNA the way that we're supposed to, and then we're not fixing it. And if we start copying broken DNA, then we have cells that are cancerous. Sometimes people refer to BRCA1 as a tumor suppressor gene because it fixes the DNA so you don't get tumors, but it's not tumor suppressor gene the way that we classically define it, but kind of, sort of. What's super interesting about this BRCA1 gene, we have already identified over 20,000 different alleles. So 20,000 plus different base substitutions that have led to 20,000 different alleles for this gene. And some of them are benign, and so we're not worried about them becoming cancerous. Some of them are quite pathogenic, and so we're far more worried about the likelihood of cancer if people have that particular mutation. So people with histories of breast cancer or other kinds of cancer in their family, they might have their BRCA1 gene sequenced so that we can figure out what allele they have so that we can determine their risk of cancer. So interesting. We have these amazing databases of these missense base substitution mutations, these different alleles or variants, and it literally shows us that we have cysteine, but now we have glycine. Or we were supposed to have serine, and now we have glycine. We had an A in the DNA, and now we have a G. So we have that specific information that we can then link to risk of cancer. Really, really amazing pieces of technology so that we can treat people to help them prevent their cancers instead of having to treat them for cancer. An important distinction to make when we're talking about these mutations is whether they're happening in germline cells or in somatic cells. If I go outside and I don't wear my sunscreen and this Texas sun zaps some of my DNA in a skin cell, that DNA could, if my BRCA1 gene isn't doing its job, lead to some skin cancer. That skin cancer on my arm, or wherever it is not going to be passed on to my children because it wasn't in an egg cell. So these somatic mutations, even if they pop up in an embryo, are going to affect only some cells and not necessarily the cells that get passed on to the next round of offspring. However, if the mutation is in an egg or a sperm, that mutation is in all the cells of the offspring, and then that whole offspring is going to be affected and will then also pass that mutation on to its offspring. So be super careful when we're talking about somatic versus germline mutations. These guys are going to affect the individual, but not the species and not evolution. Germline mutations could affect the species and therefore also the evolution of that species. Mutations in the germline could be a source of genetic variation. So here we just have a bunch of different ladybugs, or ladybirds if you are not in the United States, all the same species. These are all the same species, but this amazing amount of variation in the phenotypes, these different colorations are due to alleles, and those different alleles are due to mutation. So mutation is an amazing source of genetic variation. This is really good most of the time because it means that the species is more likely to survive if the environment happens to change. We'll talk about that a lot when we get to evolution. And we're going to wrap up here with a quick look at some of the causes of gene mutation. It's important to remember that mutation happens spontaneously and naturally and randomly all the time. But most of the time, we have those mechanisms in place to fix those mutations before they have a big effect. Remember also that we living things, we don't mutate because we need to or we want to. It just happens, and sometimes it's good and sometimes it's bad. These mutations that happen spontaneously, normally, naturally, happen at a fairly low rate. And again, we have lots of ways to fix them. There are some environmental factors, though, that can speed up the rate of mutation. Radiation, especially high energy radiation, we call it ionizing radiation, ultraviolet, X-rays, gamma rays, these can all interact with the bonds between the atoms of our DNA. And that's going to cause some breakage or some changes in the DNA. So radiation can absolutely increase the rate of mutation, and therefore we're worried about wearing our sunscreen so that we don't get skin cancer. We also have some chemicals that are also mutagens. Things like radiation or chemicals that can cause gene mutation to occur. We have mutagens like this guy, EMS, that causes a change in the nucleotide. Here we're going from guanine to this ethyl guanine. We're literally adding an ethyl group to the nitrogenous base. But what that does is it changes our base pairing. So instead of guanine to cytosine, we now have ethyl guanine coding with thymine. So we can change the complementary base pairing in our DNA, and therefore it's a mutation. Super interesting. What's crazy is that this is an ethyl group that we added from that EMS compound. It's super similar to the methyl group that we talked about when we methylate our nucleosomes so that they can be muted or not be transcribed. The only difference is that the methyl group only has 1 carbon and 3 hydrogens, whereas our ethyl group is 2 carbons and 5 hydrogens. Super interesting that they're so similar and yet have such different impacts. Another crazy thing to think about here, we can get cancer because of radiation, but also we definitely treat cancer with radiation. And there are these chemicals that can cause cancer, and then we kill cancer also with chemicals. And so a lot of the things that change the DNA that lead to cancer, we try to change the DNA of the cancer cells to kill the cancer, and it's amazing. One other source of gene mutation that I didn't add to the screen. So there are some biological agents that can also increase the rate of mutation. A super common example is HPV, this human papilloma virus that can cause increased incidences of cervical cancer because it causes mutations in the cervix. Super interesting. And we have arrived at the end of our lecture on mutations. We defined mutation, changes in the DNA sequence, changes to the DNA molecule, structural changes in the gene. We talked about those different types of mutation. We can have frameshift mutations that are insertion or deletions. We talked about all those different kinds of base substitutions, the same sense, missense, and nonsense. We talked about consequences of the mutation. Sometimes we change the protein a little bit. Sometimes we kill the protein completely. Every once in a while, we can improve the function of the protein, and that's beneficial to the species. We also talked about causes of mutation. They happen randomly, kind of all the time, but we can increase that rate of mutation with UV radiation and some chemicals, and those are called mutagens. Great work today.